o'clock. Today's talk is about my experience of receiving the Pfizer COVID-19 vaccination. I'm really happy to report that I received my vaccination a few days ago. Today I'll go through my experience, how it was here in Ontario where I live. And afterwards, we will also have a bit of a diagrammatic illustration of the various vaccines. That'll be a bit scientific, bit geeky, but I'm sure a lot of people will enjoy that part as well. So before I do that, I just need to check my streams. We are live on Facebook as well as YouTube. Anyone who has any questions is more than welcome to type in those questions on YouTube and Facebook. But I'll just go ahead, mute myself, and open the stage to the guests we have live, and they can tell us how they are and what their expectations are from today's talk. So I'll ask first my parents, then Chayaji, and then Ashish to just one by one tell us about yourselves and how you're doing and your expectation from today's talk.
Yeah, well, things are very good here under control, and the phase 2 vaccination is starting from tomorrow. So we have to register here. The registration is done first, online registration, and then we have to go for it. So it's good that we get to know the experience that Sapun had this vaccination. Though of course we come across other cases also, but from a doctor especially, then we'll have some special particular information also. Thank you, Sapun. Thank you.
Thank you so much, Dr. Sapun Singh, for very useful information you give us always.
Hello.
And we are going to get COVID-19 vaccines very soon. Thank you so much for everything, Sapun. God bless you.
Over to Chayaji.
Okay, thank you. My name is Chaya Kotnala, and I really look forward whenever I can to attend this session. I think Dr. Sapun Rawat has really contributed to this whole journey of COVID-19 so fantastically, and educating us and sharing his blog. And I'm really looking forward for today's session because it's a very important sort of chapter in our life to move on safely into the next phase of COVID-19. So thank you, Sapan, and nice seeing everybody, and over to Ashish.
Thank you, Chad. Ashish Kotial, and I have joined from Toronto. I echo what Chayaji said, and Sapan has been a great resource during last 8 to 10 months. And my expectations, or I have joined here to hear more about Dr. Rawat's experience, how he's facing after receiving his first dose of the vaccine, and also just curious to learn about the science and various platforms used by various vaccine manufacturers, what's mRNA, and those scientific terms. So it would be great to hear from Dr. Rawat and know more so that we are also prepared. I'm hearing these because of my parents. My parents are like they are in mid-70s, so I pass on this information to them also.
Oh shucks. Great, thank you everyone. So I just need 1 more minute. So Ashish, can I just ask you to just talk for a minute more about I know you do a lot of reading about vaccines and different kinds of vaccines. I'd just like you to just talk a bit about especially the Oxford vaccine, which has just recently been licensed, and what your thoughts are on the Oxford vaccine, which has just been licensed in Canada.
Sure. I'm by no way, I'm not an expert It's all what I read from various medias, digital media, newspapers, and what I'm hearing about all these vaccines, especially about AstraZeneca or the Serum Institute constructed vaccine. I'm just reading that they have an efficacy of 65%, and they are not effective on this South African variant, and that's the biggest concern. Nobody knows, and it's only the science and doctors. I'm sure there are doctors in South Africa also. They may have some reason not allowing to give AstraZeneca to South Africans, but then there are other doctors and scientists. They are saying, hey, it may not stop this South African variant totally, completely, but it may stop or reduce hospitalization or serious illness. So other than that, I'm hearing Johnson and Johnson is going to be a game changer. It's a single dose, and it doesn't require that low temperature storage. And even pharmacies, here, Shopper Drug Mart, they can start giving it, and it's a single dose. I look forward to Health Canada approving Johnson and Johnson. There are a lot of other countries who have approved. I think US approved for emergency use the Johnson and Johnson. And I'm again, by no way, I'm not an expert, but I think that our government should be proactive here and approve it so that we are not again number 25th in the line when other 24 countries have placed order and nobody gives us any preference. So that's my 2 cents, I can say. And being proactive is the best strategy here. Nobody knows what's going to happen. I was reading a newspaper again today, and there is another variant, I forgot, N440 or something, that has again shown up. So again, Dr. Sapan, I'm here to listen. The expert says, advise, you are the expert, and learn more about the science.
Thank you, Ashish. Ashish is always the go-to person if I need a bit more time to set up my system. That's great. It's so knowledgeable. One of the great things I like about Ashish is, whenever we go for a walk in the park or something, you get so much knowledge from him, because he knows so many things about so many things.
Yeah, sometimes it's hard, when suddenly you are not an expert of that field. If somebody asks me about engineering or other thing or real estate or other, yeah, but medicine, it's not. We just hear your session. You have trained us, I would say. You have given us the knowledge. Thank you, Dr. Sapan.
Great. So let me just go ahead with my talk. So I received the Pfizer COVID-19 vaccine, Pfizer and BioNTech. BioNTech is a German company. I would urge you to read up on the founders of BioNTech, a Turkish couple, husband and wife scientists who immigrated from Turkey to Germany, set up this multi-million-dollar company called BioNTech, which has done a lot of really good research, and then partnered with Pfizer to bring us the Pfizer vaccine. A really nice, you could say, immigration story as well. You could call it immigration, you could call it brain drain, depending upon your perspective. Similarly, the Moderna vaccine, one of the founders of the Moderna vaccine is from Toronto. He's, I believe he moved from Lebanon to Canada as a kid. He did his, I believe his master's from McGill University, and then he did his PhD at MIT and then set up Moderna company in Boston. And I believe quite a few of the founders of Moderna, as Ashish was telling me previously, have Canadian background. So another very nice you could say, immigration story. Immigration or brain drain, as you would like to, depending upon your perspective on this. So, I got the vaccine here in Ontario. As you enter into a group which is eligible, so we were not eligible in the first wave. The first group was hospital-based doctors. Community-based doctors came second. So I know my good friend, endocrinologist, also in the Uttarakhandi community, he is receiving his vaccine today afternoon. So we came into our community-based doctors. They came on Wednesday, and I got my vaccine the next day. I was really quick booking online. There was a online link. I clicked on it, booked my appointment. When I got there, it's a hospital-based vaccine, and that's the other thing. The two vaccines which are now licensed in Canada, Pfizer and Moderna, are both hospital-based vaccines, primarily because the Moderna is -70 to -80 degrees Celsius storage, and Moderna is more like -30 to -40 degrees storage. Moderna is being used more in the community with nursing homes, while Pfizer is almost always, except for some few exceptions, it's based in hospitals. I have to give my hats out not to just those people who have come up with the vaccines, who have developed this amazing technology and who have worked day and night 7 days a week on this, but also hats off to all the people who have in the background been organizing these large vaccine centers. It was a seamless process. I went in, I confirmed myself, I confirmed my ID, I confirmed the fact that I am eligible for it. Went through the queues, social distancing, and received it. Afterwards, initially I didn't feel very much, and to be fair, I was a bit disappointed because I always feel if you get a fever or body pains, or if you get like symptoms, and that means the vaccine is working. And I actually take a very positive view. I sometimes get patients complaining that the arm's sore, but I take a very opposite view. I feel if the arm is sore, that's a good sign. That means it's working. And so initially I was a bit disappointed. I didn't get much of a fever as I was hoping to get. But then later, the arm started getting more and more sore. And by the time it was evening, I got this in the morning, in about 10 to 12 hours, I couldn't lift my arm this much. I could only lift my arm this much. It was quite sore. So I was quite happy about that. Now, there were two trains of thought about how you should deal with it. There are some immunologists and virologists who are of the view that when you get a fever after immunization, your body is producing certain chemicals like interferons, which boost your immune response. There was one study in which patients who took Tylenol or paracetamol or crocin before vaccination had a slightly lesser immune response compared to controls who did not take Tylenol or crocin or acetaminophen before the vaccine. There was no trial to be done. What happens if you take it after the vaccine? In any case, I'm a bit biased against it. It's not completely scientific, but I do not wish to take any medication if I get a fever. So I was quite prepared that even if I'm getting chills and fevers, I'm not going to take Tylenol, because I want my immune system to get revved up. I want as many antibodies that I can make. And so the arm got quite sore. At night, I could not sleep on that side. I had to sleep on the right side because I had it on my left arm. But the same happened to my wife Jyoti as well. Jyoti also, she received the vaccine about 6 hours after I did, because we do shift pattern. One works, looks after the other one, looks after kids, and then we swap over. So for her as well, and after 3 or 4 hours, her arms started getting quite sore, and both of us couldn't sleep on one side. We had to sleep on the other side. But after about 12 hours, the arm got back to normal, and now my arm is completely back to normal. I've heard that with the second dose, in the case of Pfizer, it's anywhere 3 weeks or after, you get more systemic symptoms. So you can get more fever and body pains afterwards. I have a Fitbit, and it is the kind of Fitbit which also tracks my oxygen levels and also tracks my temperature. So at night, it did say that my temperature was about 0.4 degrees above normal. that night, but I didn't really feel it. It wasn't significant enough for me to feel any major issues. And so I'm still here. I'm still alive to tell the story. We know that Pfizer, the incidence of severe allergic reactions is about 1 in every 100,000. And typically that happens to people who already have a history of severe allergic reactions. Often these will be people who carry EpiPens with them because of, say, peanut allergy or other kinds of allergies. The particular allergen which has been identified so far is something called PEG, or polyethylene glycol. This is something commonly used for constipation. So any person who's ever taken PEG for constipation and has been allergic to it should probably not take the Pfizer vaccine, because you're almost certainly allergic to it. But this is rare, 1 in 100,000. When they had given the first 1 million doses, there were about 11 cases of severe allergic reaction. And now tens of millions of doses have been given out. In fact, all the vaccines are appearing now to be very safe. Even though there were some early signals for the Oxford vaccine and other vaccines, now that they've been given out to millions and millions of people, the safety signals are quite incredible. So, of course, you do get reports of death and after a vaccine, but it is very important to try to elicit whether that death is related to the vaccine, due to the vaccine, or whether it was just a death which would have happened anyways. Because when you give something to millions of people, just by freak chance, there will be a certain number of people who will die within that week or 2 weeks after. As an example, there was a lot of talk about deaths in very elderly nursing home, often end of care, end of life, palliative care patients. I believe it was Norway where that study came out from. But then if you interrogate that a bit further and ask what is the baseline rate of death in that population anyways, it turned out that in that population in Norway, they were having about 200 deaths per week in that population even before the vaccine. So it's very important to try to tease out when you get these kinds of news reports whether there is a certain correlation that is made, and whether the mathematical data you're given is actually different to what was happening before the vaccine or not. So that's quite important. So my experience was very positive. I'm looking forward to my booster. It's very important to realize that it takes 2 weeks for the antibodies to develop, whether it's infection or vaccine. So 2 weeks after my booster is when I'll feel I really have good immunity against this condition. Talking about immunity against the condition, I'll just have a quick talk here about the Oxford vaccine as well. The Oxford vaccine was recently licensed. Initial trial data suggested it was about 62% efficacious. In the US, recently the Johnson vaccine has been licensed as well. Both of these have something in common. They're both adenovirus vector vaccines. Unlike the mRNA vaccines, which are already licensed, which is the Moderna vaccines and the Pfizer vaccines. The Johnson vaccine also had approximately, depending upon whether it was the initial strain, the original strain, the South African strain or Brazilian strain, it had a varying efficacy rate from anywhere to the high 50s to the high 80s. Same with the Oxford vaccine, it's turning out now as well. However, I was listening to a few talks just yesterday about this, both from England as well as from here. So far, all these vaccines, Johnson, Oxford, Pfizer, Moderna, are 100% efficacious. 100% efficacious. When you look at the efficacy of stopping a person from becoming seriously ill in ICU and dying of COVID-19. And I think that's quite a big deal. That is really a big deal, because already tens of millions of vaccines have been given, and there is no single documented case of a person who completed the course. There have been cases of people who died of COVID-19 within a week or so of receiving the first dose, but bear in mind, in those patients, the vaccine had not started to have effect yet. There is not a single documented case of a person who's had 2 doses of a 2-dose vaccine who then died of COVID-19 once the antibodies had been created. Because in all of us, unless some patients who may be immune compromised, who may be transplant patients, for example, if you give the body a peek at an antigen twice, it's very likely that antigen will take you down the third time. It's very unlikely. I mean, that's how the vaccines work. How our immune system works, there is something in our immune system called immune memory. And a lot of immunologists say, and I'll just quote one immunologist, Dr. Shane Crotty, is one of my favorites. So the way he puts it is, immune memory is very similar to your own memory. If you have an incident which takes place, then you may remember it or you may forget it. We have some subconscious autonomic kind of systems which help us decide how important it is. So for example, you see a squirrel on the tree, it's not very important, you might forget that squirrel. If you are in the car and almost have an accident, you will remember that incident because to you that seemed important. Similarly with immune system, once that vaccine gives you a little piece of that spike protein to look at, in some people they remember that. In some people, the immune system remembers it for a while, but then thinks, not so sure. Is it really that important? Is it really going to be that important for me? And in some people, the immune system might just put it in the back shelf and say, don't really need this. That booster that you get is when you tell the immune system, this is quite actually important because you haven't just seen it once, you've now seen it twice. And that's when the B cells, the memory B cells, start revving up and saying, okay, we're going to keep this in the shelf, in the front shelf, because this is important. And that's how a booster works for your immune system. And once your immune memory is primed up, and that the memory of that antigen or that piece of the virus is there in the front shelf, if it comes in again, the system can get to work straight away. And that's how vaccines work, regardless of what type of vaccine it is. And so today we will have a talk as well, where we will also just go through the mechanisms of action of these vaccines. So let's get to the slightly more boring part of the talk. And this is a bit techie and a bit kind of a bit goofy, but I'm sure, I hope you'll enjoy this. Before we do, let's quickly have let's catch up with who's with us right now. Negiji from Edmonton has joined us, which is very nice. Negiji, how are you doing?
Good morning, doctor. I'm good. I was listening to you on YouTube. You're live there?
Yes. I'm on YouTube, Facebook, as well as Meet. So you're most welcome here.
Oh, thank you. I was watching on a YouTube video. It was saying it was a live streaming going on.
That's right. This happens at 10 o'clock Eastern time every Sunday. Great. So Negiji, I'll just go ahead and mute you, and I'm just going to now do a diagrammatic explanation of these vaccines. I'm just going to mute you now, and I'm going to share my screen. Great, here we are. This is a really exciting part. This is the part where I start imagining I'm some kind of immunology, lecturer and giving a little lecture on immunology. This is really exciting. So let's have a look at some basics, because a lot of people here, it's been decades since they did any kind of biology. So this is your cell. This is your cell. Just gonna figure out how to do writing. Okay, so this is your cell, and if I just use my pen, this will be the membrane. Oh, not very good with typing. I think this is gonna be that's just good for drawing, I guess. Let's use eraser. So this part here, I'm going to use the laser. Actually, it might be easier. There with the laser, there it is. Yeah, this part here is the membrane of your cell. that. And then let's make a little circle in the middle. It should be a bit better than that, but that is your nucleus of the cell. You could also call it the central command system of your cell. Every single cell in your body, almost every single cell, there are some exceptions like red blood cells, but almost every single cell in your body has a nucleus inside. And then there is one more part which is of relevance called ribosomes. So this part here is called your ribosomes. Inside every single cell of your body there is DNA. DNA is what makes you what you are. DNA is made of, amino acid sequences, and there is a process called transcription from which the DNA then turns into something called an RNA. The difference between a DNA and an RNA is that DNA is double-stranded, it has 2 strands. RNA is single-stranded. For every DNA there is a complementary RNA. So what happens is there is a process called transcription through which DNA turns into RNA. That happens inside the nucleus, then the RNA comes outside the nucleus. The RNA then goes into the ribosome, and what happens then is a process called translation, and the end product, and I'm going to draw it like a little triangle, will be a protein, and then that protein gets transported to the outside of the cell. Now let's quickly talk about the 3 main constituents of our body. When we eat something, we talk about carbohydrates, proteins, and fats. So a lot of the building blocks of our body, our muscles for example, anything which has the word globulin on it. So for example, often for the word antibody we use the word immunoglobulin, that's in protein. For our blood we use the word hemoglobin, that's a protein. A lot of the building blocks of our body are proteins. Many of the membranes in our body are fats. So for example, the outside membrane of the cell would be a fat membrane. The myelin sheaths which coat our nervous system, those are all fat based. And the third thing that we ingest is carbohydrate or sugar. Sugar is essentially the energy of our body. So for anything to function, it needs to take in sugar, and then that is converted into ATP, and that's the currency of energy. But sugar you could describe as primarily as energy. So fat is membranes, protein is building blocks, sugar is energy. And this is a very basic structure of our cell. So let's now talk about what a virus is. I'm going to make one more circle. There are various types of viruses, but essentially they contain, in some cases, a membrane or a fat layer. In some cases they don't actually. And inside there is essentially either a single RNA or a DNA. If it's a DNA, there will be 2 strands. You can have DNA viruses, you can have RNA viruses. SARS-CoV-2 is what we describe as a positive sense single-stranded RNA virus. So SARS-CoV-2 wouldn't have 2, it would have 1 strand. The interesting thing about a virus is what we describe as the obligate intracellular parasite, which means a virus is only alive inside another living cell, because the job of a virus, or the way it functions, is by hijacking your system. So it uses your ribosomes in case of a RNA virus, or your DNA, or your nucleus and ribosomes in case of a DNA virus. Sometimes even your DNA in the case of a reverse transcriptase virus like HIV to function. A virus cannot multiply without host cells. So that's what a virus is. So what happens when you get a viral infection? Well, you have your cell, and we'll quickly just draw the nucleus, your DNA, and your ribosomes. Make it very simple. So a virus comes in. In the case of SARS-CoV-2, it has spikes. These, we'll make them like triangles. And that spike has an affinity to a receptor here called the ACE2 receptor. It also uses certain other proteins called, for example, there's one called transmembrane serine protease, TMPRSS. So TMPRSS and ACE2. So it binds to that. And once it binds, it sends its RNA into your cell. That RNA then travels up to the ribosomes and creates lots of proteins. And these are different types of proteins. These are all for the assembly of this virus. The end result is that you have lots of more viruses which develop inside the cell, and they start coming out, and you have lots and lots of more viruses. So a virus comes into your cell and essentially uses your machinery to start producing more and more of itself. Ultimately, that cell dies. And that's how viruses cause damage. So what does the vaccine do? So we'll talk about various types of vaccines. And this is the really interesting part. First, let's talk about mRNA vaccines. So what will an mRNA vaccine do? Well, basically it's trying to use the same trick as the virus. So the virus, what they do is they have the first thing to do is they figured out what genetic sequence codes for the spike protein of that virus. So if you remember, the spike protein is those triangles, those little spikes on the surface. They figured out that. Then what they do is they produce a lipid nanoparticle. This is an artificial fat particle, and inside it they put the code to produce that spike. So you get an injection, your muscle cells take up that lipid nanoparticle. So they've had, this is the most difficult part of the technology. This is what took them more than 10 years. How to produce something which is synthetic, which will still get taken up by your cells. That RNA then goes inside, it goes into your ribosomes, and the spike protein is created. The spike protein is expressed on the surface of the cell, and then your immune system, and that could be your innate immune system, that could be your dendritic cells, your fibroblasts, your macrophages, your neutrophils, the cells of your innate immune system, your frontline immune system. They pick up this protein. They say, well, this is not looking right. This is something alien. This is something new. They take it to the lymph nodes and they start producing antibodies. That's a whole talk in itself. Since this is a non-medical talk, I'm not going to talk about the various parts of the innate immune system, innate versus adaptive, T helper 1, T helper 2 pathways and all that. We'll keep it simple. So this is how a mRNA vaccine works. Next, so this would be Pfizer and Moderna. Now the two vaccines which now we should be more interested in are the adenovirus vector DNA recombinant vaccines. So we're going to talk about adenovirus or we just call it here we go. So how do those work? They use, you could describe as an older platform. This platform was used for Ebola virus. So what happens here? It's quite interesting. So first, you have to figure out, well, you have already figured out the sequence which codes for your spike protein. So that you figured out. So what do you do next? Next, you have then to make what you could describe as a complementary DNA of that. So you make a DNA segment which is complementary to that spike protein. So you start with RNA which you know would code for the spike, and then you for that convert that into a DNA which would code for the spike. Now why do you do that? The reason you do that is because the virus we're going to use is a DNA virus. It's not an RNA virus. Then you take a virus which normally causes a common cold. It's called adenovirus. In the case of Sputnik, it's 2 different adenoviruses in the 2 doses. In the case of Oxford, it is a chimpanzee-derived adenovirus. And in the Johnson vaccine, it's a single dose human adenovirus. So you take that adenovirus in the cell, and that adenovirus is a DNA vaccine, so it has double-stranded DNA. Then you do some genetic engineering. First, you take out certain type parts in the DNA of that virus, which will allow it to replicate. So you want it to be non-replicating, which means you want it to be a virus which cannot multiply. Because you don't want to have an infection. You just want an immune reaction, not an infection. Next, you do a bit of cutting and pasting. You cut and you paste that spike protein into that. So you end up with a recombinant. That's why we use the word recombinant DNA virus, which is not really the original DNA virus. It has got some parts of COVID-19 spike protein, and the parts which would allow it to replicate or multiply has been taken out. Then that virus is used to infect the cell. So you inject it, that's your nucleus, that's your ribosome. Now the way this works is a bit different. This is your DNA. The way this works is slightly different. Because it's a virus, it will naturally invade the cell. That's what viruses do. And that's why this technology was with us before. The DNA moves into your cell. Now you will remember what happened with the mRNA virus. With the mRNA virus, there was no DNA, there was RNA. The RNA went straight into the ribosome and proteins were produced. But that's not what's going to happen here. The RNA went straight to the ribosome in the case of mRNA vaccines, Pfizer Moderna. But this is not a RNA, this is a DNA. DNA will go into your nucleus. So that DNA then goes into your nucleus. And so the process which took place for the mRNA vaccine, the previous one, the Moderna Pfizer, was what we describe simply as translation. Conversion of mRNA through ribosomes into protein, but that's not what happens with DNA. You need to have a process of transcription followed by translation. So the DNA gets into the nucleus, then a process takes place which is called transcription, where the RNA is made from the DNA. That RNA then comes out into the ribosome, then proteins are formed, proteins are expressed, your immune system cells pick it up. So the end result is very much the same. The only issue with this kind of technology compared to the mRNA technology is that the virus we are using is a common cold virus. When you use a common cold virus, which is human, there is a risk that you may already have antibodies against it. So let's say if you have antibodies against this vector, these are antibodies which block it, then you have a problem. That virus can get into the cell. If it can get into the cell, it cannot do this process. Which is why, and also there's another problem. When you give the first dose, you will produce antibodies both against the spike but also against the vector. And so the second dose might be less effective, the booster might be less effective. The way that Johnson got around this problem is they produced a highly antigenic single dose. So they don't have the problem with the booster being affected by antibodies to the vector. The way that Oxford got around this problem is by using a chimpanzee or monkey virus, because humans would not have natural immunity against a monkey virus. The way that Sputnik, the Russian vaccine, got around this problem is by using two different adenoviruses for the first dose and the second dose. So even if you produce antibodies against the first adenovirus the first time, this booster is a different adenovirus. That's how the various manufacturers got around this problem. Let's move next to Novavax, a protein subunit vaccine, and then finally we'll discuss inactivated vaccines. Novavax is a very interesting kind of thing because it actually this is the vaccine that is will be produced in Canada, and this is actually my favorite vaccine. And when you see the process, you will realize why this is such a good vaccine. What they do is they start in a very similar way. They start with a DNA virus. In this case, it's called bacilovirus. They take the DNA virus and then they do certain types of engineering with it. They inactivate it so that it cannot. They remove the part which causes replication, so it cannot. It can't replicate. They also put in a spike protein DNA part, which would be to produce spike protein for the COVID-19 coronavirus. But what they do next is interesting. They then infect not your body, but they infect moths which are about to turn into butterflies. So this is a moth. I'm going to make it in the shape of a butterfly. And they infect that larva of the moth with this virus. So once you've got the virus inside that moth, it goes through that same process that happen in your body, but not inside your body. It happens inside the moth. And the end result is the protein that you wanted. And then they collect the protein, and similar to the mRNA vaccines, they make in this case we don't call it a lipid nanoparticle, we call it a VLP, virus-like particle, because they make a particle which is synthetic, but it has the same size as a COVID-19 virus. So the body thinks it's a virus, but it's actually not a virus, it's synthetic. And inside there, they will put the protein, just the protein. So when this is injected, the Novavax vaccine, once it's injected into your body, this is your cell, and the Novavax vaccine is injected into the body, that the VLP membrane, it disintegrates, the proteins are in the open, and your immune system picks up the protein. It never goes into your cell, because all the work which was needed to be done in the cell, in the case of RNA vaccine, translation, RNA ribosome protein, in the case of DNA vaccine, transcription and translation, go into nucleus, DNA to RNA, RNA to protein. All of that has already happened, but inside a moth, inside another animal, an insect. all they give you is a protein, and so there is no invasion of your cells at all with the Novavax protein. The Novavax vaccine was actually the most efficacious. For the original strain, it was 96% in the trials, and about 70% for the South Africa strain. So I'm very excited for the Novavax vaccine. It looks like a really good vaccine. And finally, the inactivated vaccines are actually very straightforward. What happens with the inactivated vaccine is that you start with the virus, and in this case it's a RNA virus. So COVID-19 is an RNA virus. It's got its spikes, and then you just culture lots of it. You have live virus in the lab. That's why it's so difficult to make inactivated vaccines, because you're dealing with live virus. And what you do is you put it inside this chemical which inactivates the RNA. It puts breaks in the RNA, so RNA dies. So that essentially, now it's a bit controversial to call a virus live, because viruses actually, even philosophically and scientifically, are on the edge of life. We cannot really call them living because they cannot live without being inside another living organism. They're obligate parasites, intracellular. So for the purpose of the discussion, we'll call it, we kill the virus, although it's you could say, can you really kill a virus? It's not even alive. In any case, we destroy the RNA. However, the membrane is still there, the proteins are still there for the nucleocapsid, for the membrane, for the spike, and then you just inject the killed virus into the body, and you actually produce lots of antibodies and various types of antibodies. You would produce antibodies against the spike, you would produce antibodies against the membrane, against the nucleocapsid proteins. You would produce various types, what we describe as a broad range of antibodies to that. And because all the variants we talk about are happening in the spike, some scientists would argue that inactivated vaccine, examples include Bharat Biotech, Covaxin, and Sinovac, Sinopharm, may well be better for variants, because they attack every part of the virus, not just a spike. So that's really the talk for today. I hope you enjoyed. I know it was a bit geeky and a bit scientific, but we often talk about these concepts, and I just wanted to give you a face for this concept. Just explain what I mean when I talk about DNA recombination, about mRNA and all that. So that's it for the talk, and we can just get into a general discussion about COVID-19 and all that. How did you find the talk, everyone?
Eye-opening. Yes, I'm new in science, but it's really interesting. This is done underneath, and what's going inside our body is really incredible and scary too. When you don't know. So sometimes they say, jitna kam jano wahi acha hai. Jada janne mein your mind's curiosity increases. But I have to leave very shortly, but I have a couple of questions, Sapan, if I could ask you. You said lymphatic system works, and this it goes into your lymphatic. Suppose you don't have any lymphatic glands on one side of your body, and a lot of them are taken out, so does it mean that, I'm talking about the vaccination, should it be given into the different arm, or the doctor will decide which arm you're going to take your
That's actually a very good question, because swelling of lymph glands is a known side effect, or not effect of a vaccine, because that's how it works. It gets taken up by your immune cells, takes it to the lymph nodes. That's where you have your follicular cells, your B cells, your memory cells. I cannot quote any kind of data on this, but what I can tell you is that it may well be a good idea to use the arm which has lymphatics. That may well be a good idea. Yes, I can see why that would make sense. I'm sure if the person said, could you please do this arm because I have lymph nodes, that might be a better idea. I have a few questions which I'll take. Someone says the vaccine, ivermectin is better than vaccine. I would just answer that there is a role for both. It's going to take a long time before the vaccine can be available to everyone, and so if there is something which is got evidence to protect, such as ivermectin, then I think physicians welcome that, and if there is a vaccine, then we welcome that as well. I feel that we should not have. I don't feel there is any need for either or. I think we can have both. I have one more question, which was from Negiji. He was asking about the type of vaccine the Chinese vaccine is. That is an inactivated vaccine. So that is the same as the Bharat Biotech vaccine. Sinopharm, Sinovac, and Covaxin are all inactivated. The Chinese vaccine recently had a trial come out of Dubai, which had reasonably good results. I think it was about 85 to 90% effective in Dubai. So let's see. We're hoping to get Covaxin's results come out in the next few weeks as well. We're looking forward to that. The challenge for Covaxin and challenge for all the vaccines which are now coming out later is that they will be more exposed to variants. And as we've seen with all the vaccines, the numbers seem to go down with variants compared to when we compare them to the original strain. But let's see how well the Covaxin does. Ashish, you're muted.
Sorry, Dr. Sapan, I have no question. It was a good one to know about various platforms. But ultimately, then I think you had mentioned that what you say, mRNA vaccinations are better than others, or they are all, end result will be the same, no?
Yeah, I mean, efficacy for the mRNA appears to be better. The trial certainly showed 94% for Moderna, 95% for Pfizer. However, more real-time data coming out. There was a study the Siren study from Scotland was published just a few days ago, which showed that even the Oxford vaccine had very similar efficacy in real life compared to these vaccines. And there were differences. First of all, the mRNA vaccines were tested in hardly got exposed to variants. So that was an advantage for them. As an example, the Oxford vaccine was tested also in Brazil and in South Africa and other places. Secondly, one important part as well, the way the studies were designed, if you look at the study designs, there are certain types of biases in the study design. I'll give you one example. With Pfizer and Moderna, they measured a person getting COVID-19 when that person got symptoms. They did not count asymptomatic infection. However, that was not the case with Oxford. With Oxford, they tested the people every week. And the number that they said that got COVID-19 included asymptomatic and symptomatic both. So, to be fair to Oxford, if you had done the trial like Pfizer and Moderna, then their number may have been much more than 62%. Because we don't know how many people got asymptomatic COVID-19 in the Pfizer and Moderna vaccine groups. So I know we bat around 62% versus 95% a lot. I feel it may be much closer than that, and there may actually be no difference. And certainly the Siren study, which recently came out from Scotland, suggested that they're very similar. And what really matters is stopping people from getting sick and ending up in hospital. And for that, they're both 100% so far. After tens of millions of doses, I must add, they're still 100%, even in South Africa. And I was listening to a professor who said that, a professor from the UK, who was saying that even in South Africa, where there's 21% efficacy, which means 79% people can still get symptoms from the Covishield or Oxford vaccine. For death and ICU admission, it's still 100%. Even all those people are still very mildly symptomatic. So the point I'm making is, when you've given your body two looks at this virus and your body has produced some response, when it comes a third time, it might give you some symptoms, but the chances that it'll land you in ICU very low. In most people, the immune system is smart enough for that.
Thank you. So now another question is, and I think somebody had asked this before also, it seems now Canada would be receiving around 1.5 to 2 million AstraZeneca or the similar company's brand, other brand. And I think a lot of Canadians would receive that AstraZeneca, and there has been some hesitancy. So would there be then, who's going to decide who's going to get AstraZeneca and Moderna and Pfizer? Or it will be randomly picked up, or elderly people will get Moderna, and younger people would get AstraZeneca? Is there going to be any criteria or something you have heard about, or in your opinion?
Yeah, I think the whole concept of restricting AstraZeneca to under 65s is misplaced. I feel the efficacy is actually quite good, at par, I feel. It's a chimpanzee adenovirus. So from that point of view, there would be no natural human immunity against the virus itself. So I feel that the platform would be very effective. Now, of course, what they would do the next year for a booster, that's a different matter, because then, and that is a question for all of these virus vector vaccines. What do you do when you need a booster? Because you will have produced some vaccines for the vector as well. Will it make the vaccine less effective? So that's a question. In my mind, looking at the technology, see, so far all these vaccines have been used for emergencies, like Ebola, for example. You haven't had a situation where you had to have boosters every year or anything like that for this technology. So in my mind, that's a question.
Yeah, I think after seeing their schedule, they have, like, Ontario government has posted their schedule online, and I think 65 plus or 65 plus turn is going to be around in May or June, and AstraZeneca is going to come before that one. So I think they are planning to give AstraZeneca to 65 plus also here. Otherwise, they would say that, hey, AstraZeneca would be open to frontline or essential worker or other categories. So I think they would give that.
Yeah, plus Johnson got licensed recently in the US, and I would imagine Johnson will soon be licensed here as well. Novavax will be next in line after that, and Novavax, in fact, will be produced in Canada, as has been, because they've entered an agreement with Novavax. All three of them are fridge temperature vaccines, so they can be given at pharmacy, at family doctors. So the big push in the community where pharmacies and family doctors get involved will come with the Oxford, Johnson, and Novavax vaccines. Any questions from Negiji and Daddy Mummy before we finish? I think we've been going on for an hour now, so I think we can have some final questions.
Well, I was going to ask about one important aspect, that for how long the efficacy will remain of these vaccines. Because I was reading somewhere that some of the vaccines may be needed every 4 months. And that would badly affect the body, our system, the whole system. So that was my question, whether you have anything to say about this.
Yeah, so all vaccines have pros and cons. So out of these platforms, if it comes to safety, the safest vaccine platform, they're all very safe actually. As I said before, even with Pfizer, allergic reaction risk is 1 in 100,000. However, the safest platform is, generally speaking, the inactivated vaccines, Covaxin, for example. Even children from age of 2 months get inactivated vaccines. However, that is also the vaccine which has the biggest question mark with regards to what we describe durability of vaccination. If I give you an example between Covishield and Covaxin, Covishield Covaxin gave immune response which was better than patients with COVID-19, so convalescent patients. However, Covishield gave an immune response antibody levels which was 6 to 8 times higher than COVID-19 patients. My prediction would be that that would last for longer. And typically with inactivated vaccines, like if you look at childhood vaccines, you have to give booster at 2 months, 4 months, 6 months. And they tend to need a lot of boosters because they're mild vaccines. So it is like most things in life, you can have it both ways. You can have a very safe vaccine which is also very durable because it's a less powerful vaccine as well. So yeah, when it comes to durability, inactivated, there are questions in my mind about those vaccines. Ultimately, time will tell. Because all these patients will have their antibodies, not all of them, certain numbers of them will be part of a trial. They will have antibody levels checked. And we will find out in due course how durable the response is.
Thank you so much, Sapan. It's great help you are doing. Thank you. No questions.
There was a talk about this durability by the director of AIMS here in Delhi, and he was telling that the Covaxin may have the durability of 9 to 12 months. So that is how, if that is the case, then I would go for it. If they give me a chance, a choice.
Yes, but until we follow people up for 9 to 12 months, we wouldn't know. But it's possible. Of course, there will be 2 doses minimum. So you will be getting a booster. And so the durability of that 2 doses is what we're talking about. I think for most vaccines, if they give us 12 months durability, is quite reasonable because with all the variants happening, I think it's kind of likely that we will need a booster every year just to account for all the variants. So that's quite likely as well. Nikki, did you have a question at the end? And then we can finish the talk.
No, I just joined in between. So yeah, I got most of the point. And I have been reading interestingly about these all vaccines and following it because from last 3 months, I'm sitting at home. The province has been locked down and we have opened. The second phase is going to open from tomorrow. So it has been really a hard 3 months for us here in Alberta. A minus 50 2 weeks back. Very cold. Even it's snowing outside today. But it has been a nice winter. I went to the mountains. It has been nice. I have been reading more about the vaccines and I have been doing some kind of community services because I have enough time. So the only thing what our chief medical officer of Alberta is Dr. Dina Hinshaw. The same question was put up to her that if people want to prefer, will they get like Moderna or Pfizer or AstraZeneca, whatever vaccines? So her answer was not at this part. But because we have a shortage of vaccine over here in Alberta and they are very late on that part. But now from last week, I remember, from last week, they have started to give people of 75 and above. So they have put the birth here and according to that, it's going on. And now much better than which it was stopped for 3-4 weeks here in Alberta. Because Alberta is considered a right-wing kind of province. Here in Alberta, they say that we want to get out of Canada as well. That's the problem with Alberta. And there is a lot of politics and geopolitics, you can say, because of Alberta. In 2007, Alberta was the richest province in whole Canada. The government has so much surplus, person $700 check. And this year budget, what happened was this last week was the budget of Alberta, and this $20.2 billion deficit was there, and now they calculated next 3-year projections. So it's a very bad situation over here because of the Biden being the president of US. He stopped the very first day. He stopped the Keystone pipeline. So a lot of things going on. I don't know if you know about Alberta. It has more oil than Saudi Arabia. Alberta has largest oil reserve, but the problem here is of the oil. It's mixed with the sand, and it costs more. So when oil is $40-50, they are not making any money. But when oil go above $100 a barrel, then like it's a very golden opportunity for them. So that's the reason. And the pipeline has been, I'm listening from that pipeline from last 15 years. BC does not want it, East does not want it, US does not want it. So they are landlocked. They now only option I believe what they have is they'll have to talk with Elon Musk and get some kind of plane and get that oil to send over through the sky, because the land and sea opportunity is not there. So that is the reason. So vaccine is gonna be really good in next few months. What I believe by they have said that health. Like Alberta Health has said that by July or August will vaccinate everyone. So which is their target, and they are doing really good on that. Alberta Health is really one of the best place I have seen. They're like very good in that. Alberta Health Services, they have a good networking. They have nice budget to from last 6 years in Alberta we are not paying for health. No, not last 6 years, since 2009. Like in Ontario, in BC, you guys pay, I believe, some money to the insurance for like provincial health insurance, I believe, which is like I think something around $45-50. It used to be here till 2008. From 2009 we are not paying to Alberta Health any money. So the government still have that much of surplus, but very soon they are gonna take that family insurance from that part as well. But it has not been in discussion yet. So Alberta Health has been really a good, a public this thing for people of Alberta, and they are really doing a very good job. And that's about it.
Great, thanks Negiji for giving us that insight. Yeah, so I think yes, go ahead.
I have a question. Sorry. Somebody is sick in India with COVID, and I was going through your blogs. Your September blog talks about treatment protocol for India, and it was mild, and then this, and then you have a CAF protocol also. So can you please tell me which one should I follow? Like CAF for sure, CAF. Plus, quercetin is not available in India. So you had mentioned something bromoxylin or something. I was going through your September blog, but it's there are too many medicines. So please guide me here. Thank you.
Okay, so it depends on the risk of the patient. So if the patient is high risk, and I use the same criteria that the US uses for the use of bamlanivimab monoclonal antibody. So that would be patients above the age of 65, above the age of 55 with blood pressure, diabetes, or above the age of 18 with any comorbidity such as diabetes, morbid obesity, etc. So if the person is high risk, then I use the CAF-I protocol. The CAF-I protocol that's for India. For North America, I use the CAF-Q protocol. CAF-Q and basically Q and I are antivirals, because ivermectin is not available in here. It's not been licensed for use. In India, it is widely used. In fact, 3 of my family members who were in hospital with COVID pneumonia were given ivermectin. They turned around, so it's effective. I believe it is, anyways. The WHO and NIH still needs to be convinced on that. But so the CAF-I protocol for India is colchicine, aspirin, famotidine, and ivermectin. And the same protocol which I use for those 2 patients which I talked about last week, where I treated that 97-year-old patient with oxygen levels of 92%, and also that 65-year-old patient with severe sleep apnea with oxygen levels in low 90s, and normalize them. For them, I use a CAF-Q protocol, which is again colchicine, aspirin, famotidine, but instead of ivermectin, I use a combination of various antivirals. So that includes quercetin, zinc, black cumin oil, and vitamins like vitamin D and vitamin C. Now for India, that person would use the CAF-I protocol. That would be colchicine, aspirin, famotidine, and ivermectin. But it's certainly reasonable for them to then also use the supplements and vitamins. So I would recommend on top of that, vitamin C, vitamin D, zinc. Those are vitamins and supplements. And then depending on availability, they could also take some black cumin oil, 500 milligrams twice a day, They could also, quercetin is available. You can get it from health shops, Ashish.
Oh, really?
It is, yes. I've put the links for Flipkart, but Flipkart takes about a week, so that's too late. But you can get it from certain pharmacies. Quercetin dihydrate 500 milligrams. That is a zinc ionophore and a cysteine protease inhibitor. So it works both by blocking the virus as well as allowing more zinc to get into the cells and inhibit multiplication of the virus. So
shall I tell them to take NAV also?
Say again, Ashish.
And NAV, you had mentioned NAV for here. Shall I ask them to take NAV also?
Okay, that is what I typically do is monitor those patients, and that is my second line. When I feel that the situation is worsening, I then include other supplements, which is NAC, N-acetylcysteine 600 milligram twice a day, melatonin 10 milligrams at night, magnesium 150 milligrams, thiamine 100 milligrams. These are all omega-3 with coenzyme Q10. These are my second line. So I normally don't start with those.
Okay.
I start with the basic vitamins, minerals, and CAF-I for high risk. And then I move on to those other antioxidant, anti-inflammatory supplements if the situation seems to be deteriorating.
Okay.
I will Ashish, I have to update that protocol slightly as well. And just recently I had a patient from India as well who I had to discuss this with. So I do need to update it, and then I will call you later today, Ashish, and explain it to you, okay?
No problem. Thank you. Thank you.
Great. I believe we're done, everyone. So thank you everyone for joining us today. I hope you enjoyed the talk about vaccines. Really pleased to have received the vaccine. And I would also say that I feel it's quite important to stress that all the vaccines are good. All vaccines are really quite safe. And we have some real life data now coming out, real life data coming from Israel, coming from Scotland, which shows that even the vaccines which perhaps the trials didn't have exemplary figures appear to be very effective, in particularly when it comes to preventing death, preventing ICU admission. And so for all people, I would recommend that if you get a chance to take a vaccine, personally, even I wouldn't be too worried about which vaccine I'm offered. I would go ahead and take it. Perhaps next year when you need a booster, you may have more choices. This time around, because it's a pandemic, just take whatever you can get offered. At least from the point of view of preventing death and preventing ICU admission, all the vaccines that we have appear to be quite effective. So thank you everyone, and we'll meet again next week.
Thank you so much.
Thank you, Dr. Rawat.